Thank you, Razia ma'am. Very good afternoon to respected principal ma'am, our head Pragya madam, today's speakers, my colleagues and participants. In continuation to program, I would like to introduce our next speaker, Dr. S.V.Sai Prasad sir. It is my privilege to introduce a knowledgeable personality like sir. We are very thankful to have Dr. S.V.Sai Prasad sir as a speaker. He is a renowned personality in the field of agricultural science. Sir was born in Andhra Pradesh. He did his master from the Acharya N.G. Ranga Agriculture University, Hyderabad, and he did his PhD in 1995 from IIRR. Currently, sir is working as a principal scientist in ICAR IIRR. Sir has received many awards and honors due to his career. Some of those are INSA Visiting Scientist Fellowship, NAIP Training Visiting Scientist Fellowship, ICAR Award for Outstanding Interdisciplinary Team Research. He is also a member of Gold Medal PGDTMA by NAARM Hyderabad, Outstanding Interdisciplinary Team Research Award as a team leader, and BGRL USA BGRI Gene Stewardship Award as a member, Dr. S. Nagarajan Memorial Award from SAW BR Karnal in 2019, 21 Sukumar Basu Memorial Award from IIRI New Delhi 2020. He is also the Fellow of Society of Advance of Wheat Research Karnal, Indian Society of Genetics and Plant Breeding New Delhi, Andhra Pradesh Academy of Science. The main research area in which sir presently focus are genetics and plant breeding in durum and bred wheat. He has played an active role in developing 22 wheat varieties, 9 improved durum wheat varieties. He has contributed a lead role in the production of 23,000 acre breeder seeds of released by IIRI wheat varieties during the last 21 years, which has improved productivity and diversified wheat cultivation. He has generated approx rupees 100 crore resources through revolving funds. There is a long list of achievement of sir credit, but due to limitation of time, on behalf of our department, I welcome you, sir. Sir.
I am very grateful to the organizers to allow me to give an opportunity to me to present in this national seminar, especially Rajia and other people and the principal of the academy. I worked in Indore for about 22 years, and after that recently I have shifted to Indian Institute of Rice Research as a principal scientist, and mainly the topic of my talk is role and prospects of biofertilizers in sustainable agriculture. That means how we can use, because before Green Revolution, all our varieties are tall and having a low productivity. And because of this insufficient food at that time, they have gone for using of chemical fertilizers to increase the productivity of our croplands, mainly rice and wheat. And the plant type they have developed from tall to dwarf type, non-lodging, more tillering, and responsive to fertilizers, by which the production has been increased. At that time, it is a necessity because we are facing problem of food shortage and we are dependent on US and other people. And now, after the Green Revolution, there is a steep increase in production and productivity of mainly the staple crops, rice and wheat. And now we are self-sustained in food production. And we are having food security. And now we are thinking about nutritional security also. And at the same time, to reduce the environmental pollution, which is being done by the chemical fertilizers, pesticides, and others. Mainly the pollution is not mainly by fertilizers, but chemical fertilizers, but due to adverse use of the pesticides and weedicides. They are creating more problems. See, in my slide, you have seen different type of fruits, vegetables, and other things. Even by using the biofertilizers, we can able to improve the quality of our agriculture products. So even if you see the increase in the human population, it is going in a very steep way. So there is a major threat to our food security of the people, because we cannot experiment the crop plants by using other modes rather than chemical fertilizers, because we need the sufficient food production to keep our buffer stocks stable. So now world population about 7.6 billion, and India alone contains 1.35 billion people, which is increasing day by day. So they are putting pressure on the agricultural lands, because we are losing the arable lands for real estate and other resources which are needed for food of this huge population. Therefore, even with the available arable land, we must increase the productivity of agriculture significantly within the next few decades, because the population is increasing, the demand for food items are increasing, and so there is a need to improve the agriculture productivity. So we are depending on chemical fertilizers. At the most 60 to 70% of the area is dependent on chemical fertilizers to improve our crop production. But we know that it is damaging our ecosystem and human health because of over usage of fertilizer. Suppose if you go to Punjab and other places, they are using around 180 to 240 kgs per hectare for wheat crop to increase the productivity. So biofertilizers are one of the greatest natural gifts of our agricultural science as a replacement to chemical fertilizers, because we have different bacteria, fungi, and other organisms which are helping to improve the availability of nitrogen and phosphorus and other chemicals which are used by the plants. So the biofertilizers normally contain the microorganisms which encourage the adequate supply of nutrients to the host plants and ensure the proper development of growth, development, and regulation in their physiology. So just I want to tell to the audience that what is meant by biofertilizers, because it are defined as a biological products which are contained in living microorganisms, and when applied to seed, plant surfaces, or soil, promote growth by several mechanisms, such as increasing the supply of nutrients, because the plants need nutrients like nitrogen, phosphorus, potash, like major elements, and other micronutrients like zinc and iron and other things. Increasing these biofertilizers will increase the root biomass or root area and increasing the nutrient uptake capacity of the plant. They are also containing, when applied to the field, it colonizes the plant rhizosphere and enters into the plant vascular system and promotes growth. There is an internal effect of these biofertilizers on the plant by increasing the micronutrient availability to the plants. And at the same time, they are adding nutrients to the natural process of nitrogen fixation, solubilizing phosphorus, and promoting plant growth by synthesizing the growth promoting substances. These are non-polluting and renewable sources of plant nutrients, because the chemicals, they are polluting, whereas the biofertilizers are non-polluting and renewable sources of plant nutrients. The main sources of these biofertilizers are bacteria, cyanobacteria, and fungi, mainly. I hope everybody are able to hear my voice.
Yeah. Audible.
Okay. So what are the advantages of the biofertilizers in place of chemical? Because these biofertilizers are very cost effective. Compared to chemical fertilizers, which are now they are very much costly, because one bag of urea, it costs around 350, and one bag of DAP will cost around 1200 to 1300. So biofertilizers are somewhat cost effective. And at the same time, they are very environment friendly and eco-friendly, because they are not only prevent damaging natural sources, but also help to some extent in cleaning the plants from precipitated chemical fertilizers. And these biofertilizers are having a long-lasting effects, because they are involving the microorganisms which convert the organic minerals into simple compounds and are easily taken by the plants. Because these microorganisms normally functions in long duration. Because the main difference between a chemical fertilizer and a biofertilizer is that this is a slow releasing nutrient mode, whereas the chemicals like urea and phosphorus and potash, muriate of potash, when we are adding to the plant, they are easily available directly. But whereas these biofertilizers, there is a long duration, it will slowly made availability of these crop nutrients and improve the soil fertility also. Normally, many people have done the experiments. They have shown that they increase the crop yield by 20 to 30%, by which replace the chemical nitrogen and phosphorus by about 25%. We can reduce the utility of these chemical fertilizers by about 25 to 30%, whereas we can increase the yield also. And these biofertilizers also will provide protection against drought and some soil-borne diseases. Because against drought also, because of using of the biofertilizers and organic manures, the soil texture will be improved, and at the same time the water holding capacity of the soil will increase, whereas so that it can protect from the against the drought. Like normally many people are using the biofertilizer like rhizobium, Azotobacter, azospirillum, blue-green algae. These are some of the examples of biofertilizers which are really available in the markets, and the farmers are using these biofertilizers to improve the soil fertility as well as the production of the crop. So how we can apply? Because there are biofertilizers which are available, but there are different modes of application of these biofertilizers, either by seed inoculation with powder formulations, or dry biofertilizers are mixed with the seeds in a seed hopper, sprinkle method, whereas the water is mixed with seeds before peat powder is added and mixed, slurry method, and seed pelleting. And at the same time, biofertilizers and adhesives are applied to a slurry of seeds and coated with ground material like lime. So somehow these biofertilizers must be coated around the seeds before sowing, and the peat suspension, seed treatment or seed inoculation, soil application, at the same time seedling root dip. These are the different methods how the farmers can be applied biofertilizers to their crop plants so that they can get the advantages of the biofertilizers. There are different classifications of biofertilizers. There are different types of biofertilizers, because unless you know the knowledge of what are the biofertilizers, what are their groups, then only we can go into our crop development to improve the crop productivity for sustainable development and sustainable agriculture. So there are some nitrogen fixing biofertilizers are there, like free. There are two types of nitrogen fixing biofertilizers, like free living symbiotic and associative symbiotic. If for free living symbiotic, there is Azotobacter, Clostridium, Anabina, Nostoc. These are some of the examples of biofertilizers which can be used for nitrogen fixing. At the same time, associative symbiotic azospirillum, we can use it as a nitrogen fixing biofertilizer. In addition, there is some P solubilizing biofertilizers are also there. In that, P solubilizer and P mobilizer, because it is very P is available in the soil, but in a fixed state, so that the plant cannot able to take the phosphorus of required quantity because it is in a fixed form. So there are P solubilizers like Pseudomonas, Aspergillus, Penicillium, etc. And P mobilizers are there, BAM, Glomus, like that. At the same time, there are some organic matter decomposers are also there. Some microorganisms which can decompose the organic material which we apply to the field in the form of organic manure. There are cellulolytic which reduce the cellulose like trichoderma, and lignolytic, that means it will break the lignins like agaricus, flutorus, like this. And at the same time, plant growth promoting biofertilizers are also there, like rhizobacteria, like Pseudomonas and other things, so that they will help in growth promoting of the plants. So these are different types of biofertilizers which we can use in our crop plants. So how we can use for different crops? If suppose rhizobium, different crops are there, pulses, oil seeds and powders, we can use rhizobium. Azospirillum we can use for rice, wheat, millets, maize, sorghum, and sugarcane. Astrobacter also we can use for rice, wheat, millets. So these are the different crops where we can use different biofertilizers. So now studies are going on how the response of this biofertilizer between the genotypes or the germplasm. If suppose there are some germplasms which are responsive to this biofertilizer, so they are comparing with the effects with the chemical fertilizers. If they are getting the on-par value of in crop production, crop productivity, then they are able to identify that this particular biofertilizer, that this microorganism is suitable for that particular crop. If suppose azospirillum is there, which is responsive for rice crop, so with the use of azospirillum, they are screening the different germplasms of rice, and they are able to identify some of the germplasms which are responsive to these biofertilizers to give higher yield compared to the chicks which are already grown under the chemical fertilizers. So we doesn't want to reduce the yield of the crop plants, and at the same time we want to use the biofertilizers to improve our environmental conditions. So many experiments have been done, and they are able to identify that this is the percentage of increase by using the the biofertilizer over the chemical fertilizers in different crops. But I want to convey to this group that in the first year itself, no crop plants will show added advantage over the chemical fertilizers in the first year itself, unless you cultivate your crop plants at least for 3 to 4 years with the biofertilizer, then only it will be an organic crop and able to sustain the crop productivity. So unless you use the biofertilizers in a field for more than 3 to 4 years, then only it will convert the field into a biofertilizer, organic manure products. So here they have studied the experiments, and in wheat they are able to identify around 8 to 10% increase in yield in comparison to chemical fertilizers. Many people will feel that if you use organic manure, the crop yield will be reduced, but it will be reduced in the beginning years, 1 or 2 years, and slowly if you are able to develop the field with biofertilizers over years, so it will improve the soil fertility, soil texture, and at the same time the carbon content and moisture retention capacity, by which slowly it will perform on par with the chemical fertilizer. So that slowly we can reduce the usage of the chemical fertilizers and increase the utility of biofertilizers. In wheat, they are able to identify 8 to 10%, in rice 5%. Like that, different crops, they have seen that there is an increase in yield obtained with biofertilizers in comparison to chemical fertilizers. At the same time, the health of the human also will not be affected by using this biofertilizer used crops. So this is the plant growth promotion, different steps, how the heavy metals and release of biocontrol agents, phosphate solubilizing microorganisms, like different, all will interact with each other, and then it will promote the plant growth. Like that, we cannot say that this particular biofertilizer will increase the yield like that. There is a system where we can use all the nitrogen fixing bacteria and phosphorus solubilizing bacteria and other things to increase the plant growth promotion. The potential use of soil microbes in sustainable crop production means these biofertilizers will also improve the photosynthesis performance to confer plant tolerance to stress and increase resistance to pathogens, thereby resulting in crop improvement. Because as a breeder, because I am actually a breeder, so here main thing is that how the chlorophyll content and the photosynthesis performance will be improved so that it can grow even in stress conditions and increase the resistance to pathogens by using these biofertilizers. There are different bacterial biofertilizers are there, which having different activity like phytohormone, nutrient enrichment, improved photosynthesis performance, improved plant tolerance to stress and resistance to pathogens, at the same time increase productivity and crop improvement. because our main idea of any agriculture scientist is to increase the yield, improve the economic status of the farmer, and it must have a stress tolerance at the same time to pathogens and pests. So we must, because we must see it as a whole, as a plant, to improve the benefit, to improve the content of the beneficial microbes in the form of biofertilizers in sustaining the agriculture production. And these are the hypothetical mechanism of action of biofertilizers in the root hairs, how it is really affecting at the root level, so that the intake of the elements will be more, and the sink, source to sink, it will be increased, and we can have a higher productivity. Okay. So fungal biofertilizers for sustainable agricultural productivity, they are different biofertilizer agent like fusarium. And what is the mode of action of that biofertilizer also we must take into consideration before selecting of any crop plants to improve the productivity in comparison to the chemical fertilizers. So we must always compare the production of the biofertilizer utility plants at the same time with chemical fertilizer, because we cannot sacrifice the yield because of usage of biofertilizer. We must have the on-par production of the crop plants along with the usage of biofertilizers, because these are naturally utilizing compounds. Here recently you have seen that Honorable Prime Minister Narendra Modi ji has told that we must go for natural farming. Because of natural farming also we can able to increase the production and productivity of the crops like rice and wheat. So that slowly we must come to our natural farming. And at the same time, we must develop our plant types which are responsive to biofertilizers, because they are slow releasing biofertilizers. So we must develop the crop plants in such a way that they are sensitive to the nutrients available by the biofertilizers. They must be responsive. So these are the different mode of actions, and the microbial species which are Fusarium, Trichoderma, microrhiza fungi, Aspergillus, Penicillium, and these are the how they are functioning in the soil to help the plant to take the nutrients for their crop productivity. Suppose if you see the Penicillium, it will supply the essential minerals like P, manganese, zinc, iron, cobalt, copper, and molybdenum to the plants. Because of these nutrients, they are able to induce the abiotic stress resistance in young seedlings, and at the same time they can protect the plants from fungal pathogens. And at the same time, different Chaetomium species, it will enhance the compost degradation. At the same time, there is one Glyocladium species which will reduce the incidence of damping of disease, and Tritigium species like organic acid secretion for mineral dissolution. So all the biofertilizers have different mode of actions and which are sensitive to the crop plants. So whenever we are selecting the and developing varieties in any crop, we must see the responsiveness of the germplasm for various biofertilizers which are utilized, and what is the mode of action on which it is helpful to the plant. So in different countries, we are using the biofertilizers and the different plants. So in USA, they are using the fungal biofertilizer, different biofertilizers in different countries, and what is the crop grown. And these are the scientists which they have given that on this crop, they are seeing the advantage of biofertilizers in terms of increasing the yield, grain yield or product yield in comparison to chemical fertilizers. So always we must see what are the findings of the scientists and what are the doses of biofertilizers and what is the crop which is responsive and how we can use that in increasing and sustaining our crop production. So if nitrogen fixation biofertilizers, different scientists have given different findings. That means their Cyanobacteria, they have seen that it is fixing at an average of 20 kg of nitrogen per hectare per year in Cyanobacteria. Azospirillum is also capable of fixing of 20 to 40 kg of nitrogen under microaerobic conditions. And because of this ability of the biofertilizer to fix nitrogen from the atmosphere, and that making it available to the crop plant, the nitrogen, because of this activity, we can able to reduce the dosage of chemical fertilizers for these crop plants, so that we can increase the economy of the farmers and the profit of the farmers. At the same time, the soil conditions of the farmers' fields will be improved. Soil moisture retention capacity also will be improved. And even rhizobium in different crops like soybean and other things, rhizobium is very much useful to fix the atmospheric nitrogen in the form of nodules. Cyanobacteria, it is also a photosynthetic and can fix atmospheric nitrogen. It can also fix around 20 to 30 kg of nitrogen per hectare in submerged rice fields. In rice fields also, they have done the experiments and cyanobacteria, they increase the rice productivity around 15 to 38% in comparison to chemical fertilizers. Azolla is also being used for fixing the nitrogen, and it has varied. The potential of this azolla is varied from 30 to 50 kg of nitrogen per hectare. And if you apply this azolla in rice, it has increased the yield by 0.5 to 2 tons per hectare by utilizing this azolla species. Even Gluconobacter diazotificus is also a nitrogen fixing bacteria, which is actively fixing the atmospheric nitrogen and able to supply nitrogen to the crop plants. So these are different nitrogen fixation bacteria which can be used as a biofertilizer to improve the crop production in our crop plants. And other type of biofertilizer is phosphorus solubilizing bacteria, and even fungi is there which is phosphorus solubilizing. Phosphobacterium, it is a gram-positive and rod-shaped bacteria. It will enhance the phosphorus solubilization because normally the phosphorus is fixed in the soil. So to release the phosphorus, this bacteria, phosphorus solubilizing bacteria, will help it, and it supplies to plants to an extent of 10 to 15 kg per hectare. That means how much we are saving the chemical fertilizer of phosphorus if we can use the phosphorus solubilizing bacteria, and it will save around 50% of the cost of P chemical fertilizer inputs. Slowly we can able to reduce the usage of chemical fertilizers and increase the utility of phosphorus solubilizing bacteria. There are phosphorus solubilizing fungi is also there. Ganesh et al. Has reported that arable soil fungi like a penicillium have shown to increase the plant growth by 5 to 20%. At the same time, aspergillus they have used to increase the dry biomass of chickpea by 22 to 33% over the non-inoculated. There is one inoculated and non-inoculated. Non-inoculated where they use the chemical fertilizers, and inoculated they use the penicillium, so that we can increase we can sustain the crop production. And at the same time, penicillium, along with 50% of recommended P fertilizer dose that produce wheat yield similar to 100% P. That means we are able to save 50% of the phosphorus usage of the rice by using this phosphorus solubilizing fungi in the crop. And there are some phosphorus mobilizers are there. They are readily available in the market, which we can use as a phosphorus mobilizers. Here, certain microorganisms which will increase the uptake of P by mobilizing it from a P-rich environment to plants instead of solubilizing P. That means it tries to move the phosphorus from phosphorus-rich environment to crop plants. And a pseudomonas also, they have used, this is also a phosphorus mobilizer and it will increase the total shoot length and solubilize the insoluble P compounds. Jacobson has also showed that different amounts of external mycelium produced by this aspergillus fungi supply different amounts of P to the host plant. So at the same time, we must screen the germplasm or the segregating populations. In this, biofertilizer fields which are responsive to biofertilizers, so that we can able to select those genotypes to develop a variety with improved grain yield. This AMF also gave increased contact with phosphates in the soil by mobilizing phosphates under conditions of low P availability to produce the organic acids which are readily available to the crop plants. So these are the different types of biofertilizers, and in crop improvement, we are using these biofertilizers to screen our segregating material and the germplasm to identify the best responsive plants and to develop a varieties. At the same time, there is another major element, phosphorus, potash, solubilizing bacteria also are all there. So soluble potassium and inoculative strain so higher potassium content of the leaf was increased by 39%. That means 40% they have been increased by using this potash solubilizing bacteria. And this solubilization is carried by a large number of bacteria. Number of bacteria are there, bacillus is there, penibacillus is there, like the different pseudomonas and this type of bacteria which are useful for solubilizing the potash. And the production management of biofertilizers contain KSB can be an effective alternative to chemical. Even by reducing the usage of muriate of potash, we can use this K-solubilizing bacteria to make available the potash to the crop plants, by which we can able to improve and sustain our crop production. In addition to these major elements like nitrogen, phosphorus, and potash, there are some plant growth promoting rhizobacteria is also there. So this group of bacteria, what they will do, they colonize the roots or rhizosphere soil and beneficial to the crops. They will be like a plant growth promoters. By using this, it will be for different new inoculants with combination of different mechanisms of action leads to a more efficient biocontrol strategies to improve the cropping systems. So we can use this plant growth promoting rhizobacteria and different, these are all available in the market. And I will, at the end, I will tell why they are not able to marketize, because there are some problems. At the same time, there is some zinc solubilizers are also there available in the market. Zinc can be solubilized by microorganisms like thiobacillus, Saccharomyces, and these are used as a biofertilizer to soluble a fixed micronutrient like zinc. Because nowadays, the biofortification of the rice varieties or wheat variety, biofortification of crop plants is mainly focused on zinc and iron content. By improving the zinc solubilization and zinc availability to the crop plant, the zinc content of the rice and wheat can be increased. It can be used as biofertilizer for zinc, and in soils where native zinc is higher, with insoluble cheaper zinc compounds like zinc oxide, zinc carbonate, zinc sulfate ke alawa, we can use the zinc oxide and zinc carbonate also, and different zinc activators are also there, so that we can use these biofertilizers to crop plants, so that we can reduce the environmental pollution by using of the chemical fertilizers. So now at the end, we want to know what are the rejuvenation strategies of biofertilizer for sustainable agriculture development, economic development. To maintain the sustainable agriculture production and productivity, what are the strategies to rejuvenate for the biofertilizers so that the usage of the biofertilizers can be increased? That means we must identify and selecting efficient locations and what are the crops are responsible, which type of soils, and specific strains of N fixation. Because my friend Dr. S.K. Sushil Sharma has developed many bacterial strains which are useful for soybean during his stay in 15 years at Indore. So like that, microbiology scientists are already developing some specific strains of N fixation, P and zinc solubilizing and absorbing to suit different agroclimatic conditions. We must be able to identify the strains and which are the crops are responsive, and then which are the varieties which are responsive, and we must see in the segregating population which are the genotype plants are showing some sensitivity to the biofertilizer, so that we can able to select those plants to improve our production and productivity. At the same time, different cultures are available between countries. So we must able to share, because USA and other countries are also doing on biofertilizers. So we must exchange our cultures, because in India also we are having some cultures and local cultures, and different countries are. We must exchange the cultures of similar climatic conditions. We must see that what are the countries where our similar climatic conditions are there, and evaluate their performance for a better strain for a particular crop, as well as checking the activity of culture during storage. They must not go for mutants. Natural mutants must not be there, so that we must be able to maintain the culture, and we must see which are the crops are responsible for that biofertilizer, and then we can able to develop the varieties in those crop plant for that particular culture. And at the same time, we must develop some suitable alternative formulations like liquid inoculants, granular formulations, that is, so that it must be easily utilized by the farmers in the field. It's not that there must not be a complicated procedure to use the biofertilizers in the fields. We must be able to develop suitable alternative formulations, granular formulations for all bioinoculant to carrier-based inoculants, standardizing the media, method of inoculation for the new formulations. At the same time, microbiologists are very important for identifying the strains and production units to monitor the production of the biofertilizers. And we must develop cold storage facilities and production centers to maintain the culture activity. At the same time, we must train the people on production and quality controls to the producers, and rendering technical advice and projects to manufacturers. At the same time, organization training to the extension workers and farmers to populate the technology. It's not that we have a good strain, biofertilizer is there. Unless you train the farmers and the extension workers, you cannot able to populate the technology. You cannot able to populate your product, biofertilizer product. At the same time, we must disseminate this information to mass media, publications, and bulletins, so that it will be easily available in the awareness can be increased using of these biofertilizers, and what is the advantages of these biofertilizers in the fields. Even now also, and presently, many of the farmers are using the biofertilizers, even in pulse crops, even in soybean and other things. Many farmers would use rhizobium. Different cultures are available, and government is supplying to the farmers at a very reasonable rate to apply this rhizobium, so that the nodulation will be increased. At the same time, the productivity and production of the soybean crop can be sustained. But why it is not getting popular? Why we are not able to replace? Because we cannot able to replace complete chemical fertilizers, but there must be a balance between, and the utility of the biofertilizers can be increased. Because chemical fertilizers, immediately it will affect the crop yield. If suppose, in a field, if some yellowness is seen, then immediately people will advise go for one spray of urea or one dose of urea, so that the plants will be responsive and the tilling will be improved. So in that way, we must have to balance the chemical fertilizer, usage of chemical fertilizer and biofertilizer, and slowly the utility of biofertilizers should be slowly increased. Main thing is that the main problem of the biofertilizer technology, why it is not getting popular, is this unavailability of good quality carrier material and lack of qualified technical person production units. Many products are coming to market, but main thing is that good quality carrier material is to be there. And at the same time, environmental concerns like seasonal demand of biofertilizers, simultaneously crop operations, and short span of sowing and planting in a particular locality. So when many people will say that get one available of one bag of urea, okay, it will be suitable for the crop to immediately respond. But these biofertilizers will take some time. So there is a environmental constraints, unawareness of the benefits of technology among the farmers due to problem in adoption of these technologies, different methods of inoculation, and no visual difference the crop growth immediately. Because immediately if you give a dose of chemical fertilizer like urea, DAP or something, it will be a difference in growth crop growth immediately so that you can respond. Same as allopathy and homeopathy. Allopathy, if you go immediately, if you go to a doctor for a headache or some other this thing, they will give some medicine. So immediately you will see the difference. But in a homeopathy, it is very good for health, but at the same time, it will have a long duration. So there must be a balance always. We must make a balance between the usage of biofertilizer and chemical fertilizers, and slowly, and at the same time, marketing constraints are also there. Non-availability of right inoculant at the right place at the right time, lack of retail outlets and the market. At the same time, here we are dealing with a living organisms like bacteria, fungi, and other things, and we must be very sensitive in storing those products. Whereas chemical fertilizers you can store at any place you want. So these are the unavailability of suitable strains, unavailability of suitable carrier, lack of awareness among the farmers, what is the dosage to be used in which crop, what is the dosage to be used, and skills and technical know-how and environmental concern. These are some of the drawbacks, but slowly we can able to overcome these barriers and make the biofertilizers popular among the farmers so that our environmental pollution can be reduced. At the same time, the soil conditions can be improved, soil texture and moisture retention capacity can be increased. Some of the new biofertilizers are coming into the market, and because whatever the products are coming into market, the crop improvement scientists or the personnel who are involved in crop improvement should test these biofertilizers to their croplands and to different segregating populations to be able to identify the plants which are responsive to these new biofertilizers and in terms of their grain yield or production productivity. Among the fungal biofertilizers, mycorrhizal biofertilizers greatly appeal to the agro industry mainly due to its versatility and use of environment-friendly technology. Still, the research is going on, supported by advanced technology to enhance the biofertilizers use worldwide, as well as its profitability for the small and minor. Because mainly 80 to 85% of the farmers in India is of small and marginal farmers. So we must develop the technologies, whatever is available in the market, for targeting those small and marginal farmers so that the biofertilizers utility can be enhanced so that we can sustain our crop production in an efficient way. So concluding, I want to, in the end, in concluding and future prospects of the biofertilizers, as a boon for farmers, biofertilizers being essential components of sustainable farming play a vital role in maintaining the long-term soil fertility and sustainability. So we must educate the farmers and all the personnel to use the biofertilizers in place of chemical fertilizers at a larger extent. And these biofertilizers would be a viable option for farmers to increase productivity per unit area in organic farming from an era of prosperity and clean environment. So we can able to utilize these biofertilizers in a proper way in the croplands. Currently there is a gap of 10 million tons of plant nutrients between removal of crops and supply to chemical fertilizers. That area we must able to monopolize by using the biofertilizers, because we are not able to supply the chemical fertilizers to the total cropped area. So whatever the gap is in between, we must monopolize with biofertilizer usage in those areas to increase the production productivity. In context of both the cost and environmental impact of chemical fertilizers, excessive reliance on the chemical fertilizer is not viable strategy in long run because of its cost, both in domestic resources and foreign resource exchange also, because we are importing the chemical fertilizers involved in setting up fertilizer plants and sustaining the production. We cannot able to sustain the production by using these chemical fertilizers. So biofertilizers are products that are likely to be commercially promising in the long run. So slowly we must aware the farmers and whoever is involved in agriculture, they must be aware by using the biofertilizer so that it becomes available to producers as well as farmers. The use of biofertilizers in India will not only have an impact on sustainable agriculture economic development, but it will also contribute to a sustainable ecosystem and the holistic well-being of the country. Because of using of the biofertilizers, we can able to sustain our agriculture production slowly as an alternative to chemical fertilizers, and it will also contribute to our sustainable ecosystem. That means our environment will not be polluted, and the holistic well-being of the country. So at the end, thank you for the organizers, because if we use the biofertilizers slowly and consistently, we can improve the crop production and productivity and sustain our crop production, and we can able to have these good vegetables, fruits, and crop plants by using the biofertilizers. Thank you very much. And once again, I want to thank you, the organizers, who has given me this opportunity to present this talk. Thank you very much.
Sir, your presentation was very informative, encompassing the entire concept on biofertilizers. I would like to open the session for discussion. Are there any questions which our participants wants to ask, sir? Please raise your hand. We'll unmute you from this side. Sir, there's Nishant Sirse. Can we unmute Nishant Sirse, sir?
Hi.
Yes, Nishant,
Thank
sir,
you.
you can ask the question.
Yeah, thank you very much, Dr. Vasai, for such an informative session. So I saw in your presentation that one of the reasons you state for biofertilizers are not so popular is because they don't have an immediate effect. And I think this is one of the main reasons for farmers not to take up this technology to use biofertilizer, because we are all aware about the situation of farmers. So they want something which has to be immediate effect. My question to you is there any way, you have shown a slide that shows there are different yields as for different crops. It's better than chemical fertilizers and they are higher efficiency compared to them, but it is different. Is there any way to increase the efficiency of biofertilizer for one particular crop, particular soil? Is there any technology from different from microbiology that could be done for increasing the efficiency? This is my question. And the second question, I think you also said about the shelf life. I think farmers are also worried about what are the shelf life, the fertilizer they are getting, what is the shelf life they have?
Thank you, Nisha. It's a very good question, but mainly the farmers, we must aware the farmers that using the biofertilizers in a long run, it will have an impact on the crop. So it is not that in the first year by using the chemical, using the biofertilizers, they will have a good yield. Over 3 to 4 years, then it will be responsive as a chemical fertilizer, because by that time the soil conditions will be improved and the strains which we are using in those organic manures, that will help the croplands to immediately show the performance. At the same time, the shelf life you are using for the these fertilizers, it can be better explained by my friend Dr. S.K. Sharma, because he is a microbiologist, so that how to maintain this shelf life so that it will be useful to the farmers.
Dr. I'll explain on this aspect. My first question, I will also explain on the first part. See, microbes are living in the soil, where organic carbon is very, very important. Number one. Number two, our soil has been contaminated, polluted like anything. So once you give the microorganism in the first year, you will not get the response because they will be targeted by the environmental factor, that's to pollution. So unless, that is why it will take some time, 2-3 years, so that it will neutralize the pollution of the soil, and then they will act. So there are two aspects. One is organic carbon, that is very, very less, and another is acclimatization of a particular kind of microorganism in the soil, then only you will get. The third explanation for this is, always chemical fertilizer is better than the microbes in terms of response. In terms of response, it is in only in terms of response to the crop, but microbes are not only providing nutrient. Dr. Saab has talked about the biofertilizer. Now the concept has become biofertilizer, biostimulant, and biopesticide. There are three categories now. Earlier it was biofertilizer. Now there are three categories, and in one microorganism, one microorganism can do all three aspects. So microorganism and strain specificity between the plant and microbe is very, very important. So for any plant, you need a particular type of strain. You cannot use any other microorganism or any microorganism which are not useful, because plant microbe interaction is very, if they are not interacting, if the microbe is not rhizocompetent, if we are not colonizing, it will not respond. That is why we are getting differential response, not in the form of yield only, it is in the form of the other parameters like chlorophyll content, growth parameters. So this is the first response for your question. The second, shelf life. Now what we are doing, we are using carrier based fertilizer, particularly talc based, lignite based. Now we are moving toward the liquid. And not only the liquid, we are also going toward the nano formulation. So nano formulation is also very important. But to increase the shelf life, nowadays several chemicals are being used to the shelf life. But it is also what kind of microbes you are using. If you are using bacilli, they are spore forming. If you are using pseudomonas, they are gram negative, they are not spore forming. And they are also mesophilic organisms. They will survive only at the 30 degrees centigrade. So it is also depend on the what kind of microorganism you are using for formulation. Then based on that, you have to develop a strategy. And our scientists today, we are developing a strategy how we can increase the shelf life of the plant. Although it is today, our government is also requiring for 12 months, only 12 months. Not only that, because microbes are not surviving, sometimes what is happening in the industry and others, when you are culturing our microorganisms, we are losing the trait of the microorganism. Yes. So how we can maintain, suppose a microbes is phosphate solubilizing. If you are culturing for a long period of time, the microbes will lose that trait. So there are many aspects. That is why Dr. Sai has emphasized many aspects, not one, strain difference, carrier-based formulation. So we have to develop a good carrier-based formulation. That is why India is now pursuing for the commercialization, developing regulation so that we can develop a good microbes with good shelf life and good productivity. So there are still constraints, and our farmers are also not aware of much. That is so popularization, advertisement, these are also very important. Our shops are not acquainted. People are not now today. What the people, everybody is acquainted with the coronavirus. Why coronavirus is so? Because it is affecting the each and every one. No, even coronaviruses was known in the animals, in the dogs also. Dogs also, when you are keeping your pet, you need to immunize your dog with the coronavirus vaccine. Now we are talking about the human, because coronavirus is also microbes. So now government policy is changing every day. Every day you are following corona appropriate behavior. Why? Because microbe is changing. And this microbe is so infectious, that is why we are so alert, our government is so alert, but we don't have such policy for improving the biofertilizer productions. And there are, I'm not publicly saying here that some of the mafias are also there. They don't want that to popularize, but the UNs are now imposing that you should popularize the biofertilizer so that you can achieve the sustainable development goal, particularly SDG 1, 2, 3. So these are the 3 goals which are very, very important to achieve by 2030. So to my friends, it's where there are very challenges an opportunity too. That is why everybody is working on this aspect. Thank you, my friends, and we will discuss later.
Thank you very much, Dr. Sharma and Dr. Vasai,
Thank you,
for the
Sharma ji.
detailed explanation.
Thank you
very
very
much.
Sir, Sharma sir and Sai Prasad sir. Are there more questions? Sankrita Shankar Gaonkar. Sankrita, can we unmute her? Yes, Sankrita.
Yes, am I audible now?
Yes, you are audible.
Sir, thank you for such a nice talk, first of all. And so my question is, does use of particular biofertilizer varies with the phenology of the crop plant?
What is it?
Again?
Is it not clear?
Question,
So-
kindly repeat the question.
So, does the use of particular biofertilizer varies with the phenology of the crop plant, depending on the growth?
No, it is not varying of the phenology, but between the germplasm or the different lines. Suppose in rice crop, if suppose there are 500 germplasm is there, there will be a variability of the germplasms in the sensitizing the rhizobium, the biofertilizer, because they are responsive. We
Yes.
cannot say that all the germplasm available are having the same type of reaction to the biofertilizer, because some germplasms will be more sensitive to biofertilizers and some are not responsive to the biofertilizers. So there will be some genotypic level, there will be a variation. So we must able to identify those genotypes which are able to sensitize the rhizobium and able to interact and produce more yield.
Dr. Shah, I want to supplement with you.
Yes, yes.
See, today what is happening that you cannot use all microorganism for all micro. And that is why today we are saying that the native microorganism. What is native? Native means if you are growing soybean, if you are growing soybean, you need to isolate microorganism from that soybean.
Yes.
Why I am telling this? Because they are rhizocompetent. What is rhizocompetence? Rhizocompetence is nothing but they will colonize our rhizosphere, the root surrounding area, very quickly. If they will not colonize, the plant will not respond.
Yes.
That means what? Plant and microbe is not interacting properly. Once interaction is not there, another example in this way, sometimes what is happening in the human beings, one injection doctor is putting to the one person, he is not reacting. But with the same injection, another person is reacting. So that is what is called reaction.
Yeah.
So reaction between the plant and microbes is very, very important. And that is why today, not only today, but we are always speaking that strain should be very specific. For one example, I am giving you on soybean. I was in soybean. We have isolated many bacteria from the soybean rhizosphere. What we got out of 400, 500 screening for phosphate solubilization and phytate mineralization, we got one bacteria, and that which called Bacillus aryabhatae. So Bacillus aryabhatae, we have screened out of 500. We also screened some other also, Bacillus other species. But that time this aryabhatae was not known. Aryabhatae was discovered from the stratosphere of the rockets. Rocket were going, they sampled soil sample from the air sampled from the stratosphere, and from there this bacteria was discovered from the India, and it is very similar to Bacillus megaterium and Syphilis. But this is a new bacterium, and that we have given this cultures to the Shivaji, who was in CCMB that moment of time. He sequenced and then confirmed that this is a bacterium. It is just aryabhatae, and that is why we got one new bacterium from the soybean rhizosphere. And today also we are using in Soybean Institute. I am also part of that. So this is one bacterium for soybean and wheat, very, very important, not only alone, but when we are putting mycorrhiza with that, that is also interacting very well. You can visit to the soybean institute, and Dr. Mahavir Sharma is there. One of my colleagues is there. You can talk to him. Dr. Ramesh is there. You can talk to him, and you can get the knowledge how the crop is responding, particularly soybean is responding with the Bacillus aryabhattai and along with the
And now they are commercializing and giving training
Yeah.
to students also.
yeah.
In that,
That is why I'm saying a particular strain is very, very important. Sometimes just 2 days back, I got one research paper, and he was not mentioning that which from the China, he was not mentioning then which Bradyrhizobium he was using. My first question was, which strain you are using? First mention. Then only you can say that this is the Bradyrhizobium japonicum strain isolated from this region, and this is functioning on that. Because he was not working on only on the plant interaction, but he was also working on the microbial community shift by introduction of that microorganism. So friends, this is also very, very plant microbe interaction is very important. You have to choose, like medicine we are choosing, we have to choose right microorganism for the right plant.
right.
And sir, actually,
Yes.
when I was working with AM fungi, that is microbial fungi, so that time I noticed that using consortium of microbial inoculants is more efficient than using single inoculant. So, what you say on this? That using consortium is better than using a single inoculant, right?
But madam, we are not able to hear.
Can you repeat your question?
You are unmuted. Unmute yourself.
Yes, sir. So, am I audible now?
Yes.
Yeah, no.
Okay. Sir, when I was working with AM fungi, so I noticed that using consortium of microbial inoculants is more efficient than using single inoculum. So,
It depends on
what,
the situation, madam.
okay.
Oh,
So it is
okay.
not always correct to use the consortium.
Okay, okay. Should I reply, Doctor?
Yes, yes, sir.
Now the question is why consortium is very effective, not single. Sometimes singles is also very, very effective. But consortium, why consortium are so
It is a
effective?
group.
It is a group in rhizosphere or in the soil. It is not the one microorganism which is playing role for the growth and development of the plant. Have you heard the name of AM supporting bacteria?
Yes, sir.
Have you heard?
There are.
So sometimes what is happening, the bacteria which you are choosing, it is supporting growth of, or it is supporting growth of AM fungi.
Yes.
And why if you are inoculating both, they are acting in a good way. For example, if you take the phosphorus and using Bacillus as a phosphorus solubilizing bacteria, and AM you are also along with that.
Yes.
So what is the function of both? One is a let me explain. One
Yes.
is a phosphorus solubilizing, another is a phosphorus mobilizing. You know what is the difference between these two? The
Yes.
difference between these two is the phosphorus, fixed phosphorus available in the soil is the governed by the bacillus present in the medium. And who will transport? Because phosphorus is an immobile nutrient. Immobile means it is not available to the plant. It will not move from here to there. So they need some conduit. Conduit means some pipe. So what is happening? They play a very synergistic role. One is solubilizing, another is capturing and mobilizing and giving to the plant. So this kind of synergy, if there, plant will respond in a very good way. And if kind of synergy will not there, suppose if you are not using phosphate solubilizing bacteria, you are using some other trait, they may not work. But nowadays, what is happening in mycorrhiza also, they are using ammonical nitrogen from the bacteria, and they can use in the transport. So that is one paper was there in one of Nature in 2005. They have proved that they are also mobilizing nitrogen. So in the presence of bacteria, suppose Penibacillus is there or Rhizobium there. You might be knowing that Rhizobium, mycorrhiza, soybean or legume plant
Yes.
tripartite symbiosis. This tripartite symbiosis is very, very important. Not, I have given one example, but there are many in the abiotic stress condition. Trehalose is the one which is produced by the bacteria, utilized both by the bacteria and the fungi for conferring tolerance to the abiotic stress. So these are the, if they are compatible to each other, they will help you. If they will not compatible to each other and their interaction will not be proper, they will not help. So there are many reasons for the compatibility. One, first microbes should be compatible, then microbes and plants should be compatible. Then only they will respond in a very good. And in that case, your soil is also very, very important. What kind of soil you are using? There are different type of soil and microbes are behaving differently. Suppose phosphate solubilization in black soil and phosphate solubilizing in the acid soil of the Pantanagar or anywhere else. They are two different and the substrate will be entirely different. So there are many factors which are involved for the good plant microbe interaction. So only microbiologist, pathologist, and scientist, if they work together, they will give you something. Not only that, breeder. If they develop a good breeding material, they will colonize good kind of microorganism. They will harbor good kind of microorganism for them. That is why breeding and then AICRPS trial, multi-locational trial is very, very important. So this is I hope you are.
Yeah. Thank you, sir. Yes, sir. Thank you, sir.